I always feel I'm too short, so I'm just going to be here. So, first of all, thank you for the invitation. I'm really happy to be here with you. I hope the next time I'm in Alberta, I can see more beekeeping action. So I work at the Honeybee Research Center, University of Guelph, and I've been very lucky to collaborate closely with the Technology Transfer Program of the Ontario Beekeepers Association, with the Ontario Animal Health Network and the Ministry of Agriculture to discuss ways to control Varroa destructor and apply that to bees in the province. So I thought about it was a good idea to see where this IPM, Integrated Pest Management approach, comes from and how it relates to honeybees. So after the Second War, with all this green revolution, agrochemicals became available in a massive way to farmers.
Yeah, that one is just for the video, sorry.
Oh, I'm so sorry. Okay, then I'll be here. Farmers had these agrochemicals available. They were being produced in a massive scale. And these researchers from California and the Cotton Belt in the US thought about long-term consequences of using chemicals in this way. For example, they thought about pests developing resistance. They thought about environmental consequences, such as polluting soil and water, and also safety for the farmers that were using these chemicals. So they thought about why not stop thinking about eradicating pests, but controlling the pests, and know in which levels they would harm the crop and the pocket of the farmers. So they thought about using all the tools that they had available to control the levels of the pests, and only if those levels were above the threshold, they would treat with chemicals. And this approach became popular with other models, animal models, including beekeeping. And one of the best examples is with Varroa mites. So then the rest of the presentation, I'm going to focus on Varroa destructor and beekeeping operations, and the integrated pest management approach that is recommended by the Ministry of Agriculture in Ontario and the tech transfer team from the Beekeepers Association. And I'm going to talk about some of the involvement of the University of Guelph to work together towards this approach. So the integrated pest management involves various strategies. The first one is awareness. That means know your enemy. And I think Varroa destructor, for all of you, does not need an introduction, but it's always good to remind ourselves what we're working with and why this Varroa destructor. I guess beekeepers, maybe we talk more about Varroa than we talk about bees. And that is because it's one of the most damaging parasites that beekeepers have to face. It came in the 90s to North America. It's here to stay. And it causes various things at an individual level for honeybees, and it has consequences for colonies. For example, it reduces the longevity of the bees. There are studies that have shown immunosuppression. It vector viruses, as you heard in the presentation. For example, deformed wing virus and black wing cell virus. These viruses not only are transported or vectored by the mites, but they can also reproduce in the mite. So that means that if you have high Varroa levels, there's a high probability that you will have high virus levels. If you have a weak colony with bees that don't live that long, that they are infected with the viruses, that they have deformed wing virus as a symptom, then you will have colonies that will produce less honey. And in North America, it has been pointed out as the main cause of overwinter mortality. We've done some research at the Honeybee Research Center in the last years. And these are some of them. For example, we parasitized bees with Varroa destructor, and then we did an RNA-seq analysis. That means we saw, compared to the control, how many genes were affected, if they were up and down regulated, and then we saw to which biological pathways they were associated with. And we found something that were not surprising, There were immune pathways that were affected with up and down regulated genes. Energy metabolism was affected. That was not surprising because Varroa feeds on the fat body. But we also saw some interesting things, such as neural processes were affected. So we digged a little bit more, and we used some genes that our lab have been working with, for example, neurexin, neureligin, acetylcholinesterase. These genes are linked to, or they are involved in neural processes, such as memory retention, learning. All these things are important for the survival of bees, because without these, they wouldn't be able to orient themselves, to forage, to come back to their colony. So we kept going, and we parasitized the bees. Then we trained them to associate another with a reward, just like Pavlov's dog. Remember that dog that they rang a bell and he was already salivating, waiting for the treat? We did the same with honeybees, and we found that the bees that were parasitized with Varroa destructor, they didn't remember as much as the control. So the proportion of bees that memorized was lower. So this is of interest because that means Varroa destructor can also be affecting some behaviors. So we're looking into that, and we're still working on that. Also, we said, why not? Let's see how multiple stressors affect the bees. So we parasitize bees, and then we treat them with an insecticide, clothianidin. And we saw that the bees that had both stressors, they groom less, and there were a less proportion of bees that groom intensively. Grooming behavior is a trait that is associated with the control of Varroa. So that also means that the bees that are parasitized would have less tools or less ways to defend themselves against Varroa destructor. So these are some of the examples that we've been working on with Varroa at the University of Guelph. So I guess this slide needs no introduction, and you already saw that in the previous presentation. But it's always good to know your enemy and remember what's going on. So you already saw that. The queen will lay egg, a little larva will hatch from that egg, and before the larva is capped to start its development as a prepupa, a female mite will go inside. Approximately 60 hours after, she will lay an egg that it will become a male. So if there's only, hypothetically, only one female mite in that cell, she will mate with her own son. Then once she's fertilized, she will start laying eggs that will become females. So approximately every 30 hours, she will lay an egg. So that means that when a worker bee finishes her development, there would be around 3 or 4 female mites that will be emerging from the cell with the worker bee. If it is a drone cell, then because it has a little bit more time to lay eggs, then you'll have 5 to 6 Varroa mites on the drones. So Varroa, they tend to prefer drones more than worker cells. So imagine if a queen bee lays 1,000, 1,500 eggs a day, and if those larvae are parasitized by a Varroa destructor, you can multiply that by 4. Of course, not all the Varroa mites will be viable or mature, but that gives you a sense of how quickly and how much Varroa destructor can develop in your colonies throughout the season. And that brings us to how Varroa destructor is related to the growth of honeybee colonies. In North America, we are all familiar with how our colonies work. So you can see in the spring, the adult bees are the dotted line. So you can see that in the spring, there's not a lot of adults. They are coming from overwinter. But once the queen has the resources and they have the go for the year, they have nectar, pollen. Beekeepers come back from Florida and start working, then they know, and the queens will start to lay eggs. the bees will finish their development, and you'll see a peak of honeybee population in the summer. That adult bee population will go down in the fall, and then you know these bees will be the ones that will overwinter. So along with that, we have the brood production that would be mostly in the summer when there's resources for the bees to breed or tend to their brood. But inside these cells, you will have Varroa destructor. So you will see a peak of Varroa destructor when all the brood emerges from their cells. So that's why the fall is a critical time for beekeepers. If you're not familiar with your Varroa destructor population, it's always good to monitor throughout the year and get familiarized with the levels of Varroa destructor, and you will see the highest numbers probably in the fall. Now, if you monitor for Varroa destructor adults, just keep in mind that there will be Varroa destructor inside the cells whenever you have brood. If that makes sense? Okay, let me know if I don't make sense. So this brings us to the next step of an integrated pest management approach, which is monitoring. So the Ontario Ministry of Agriculture, they recommend basically 3 methods, although there could be more. These methods or details, the beekeepers, they can find them in the Ontario website or in the Tech Transfer Program manual. One of them is the alcohol wash. Basically put alcohol, half a cup of honeybees from the brood chamber, shake them 3 minutes, and then count the number of bees that are in the wash. Then you can calculate the number of mites per 100 bees. And very similar to that, we have the ether roll. You have a jar, half a cup of bees, spray them with ether roll or a car starter. Then you roll them and check how many bees are on the walls of the jar, and you can have an idea of the number of mites per 100 bees. Then the favorite methods of the Honeybee Research Center is the sticky papers. You can buy them or you can make them yourself. It's a very sticky and laborious job, but you can do it. So it's basically putting Crisco in a folder file. You can put some squares to facilitate the counting. And you have to calculate the number of drop mites per day. So we recommend to leave them for 72 hours. You have to be very exact on when you put in and out and when you take out the folder or the sticky paper. And then you'll have a lot of fun counting mites. And then you have an idea of the number of mites per 24 hours. Ontario have these thresholds of number of mites per 100 bees. They are available to beekeepers in the website. Again, this was a study that was done in the province, so the thresholds could change depending on where you are. But you can see that depending on the monitoring method you use, you have a threshold for the spring and for the fall. If you are below that threshold, you don't need to treat. If you are in the threshold or above that threshold, you have to treat. In the fall, I've seen colonies in inspections that have 120 mites per 100 bees. If you see this, you are in big trouble. So this gives you an idea of what you need to do. And you have to be cautious about these thresholds as well and consider what methods are you going to use throughout the year to control Varroa destructor population. For example, if you are a honey producer, what you want is strong colonies with a high population of bees. But that means you'll probably have a high population of Varroa mites at the end of the summer. But if you are a queen producer or a nuke producer, that means that throughout the year you'll probably be dividing your colonies and interrupting the cycle of the reproduction cycle of the honeybees. So you probably have less number of mites at the end of the season. So you have to consider all the tools you have, your type of operation, and always monitor your mites and never underestimate them. Keep a close eye on them. And also, at least, well, depends also on the number of hives you have in your yard. It would be the number of colonies that you'll be monitoring to have an idea of what's going on in your operation. And that brings us to the next strategy of the integrated pest management approach, which are cultural methods. For honeybees, we have these tools. Unfortunately, we don't have biocontrols so far, but we do have cultural methods, and they are basically ways to interrupt the cycle of reproduction of Varroa mites. So if we know that Varroa mite cycle is completely related, linked, associated, attached to a honeybee cycle or brood cycle, so any way that you interrupt that cycle, you'll be doing a cultural method. I'm just going to mention some examples, but you can find more details in reliable sources. So one of them, for example, is putting a drone frame inside your colonies. We know that Varroa prefer drones, so they will go to those cells. You have to take the frame out before they hatch, because if not, you'll have a Varroa production, not a honey production. So just keep in mind that and mark your calendar and be aware that you are doing that. There's different ways to kill the mites. You can freeze the comb, you can scrape it, just make sure you kill the mites. And you put it again back to the colony. Dividing your colony, requeening. There are some techniques even that they cage the queen to interrupt her laying eggs. So there are different methods, but they are all based on the same principle. Just interrupt the cycle of Varroa mites. Control or cultural methods, if you do that, it does not mean that they are a replacement for treatment. You are using all the tools you have to reduce the mite population. Then we have the first line of defense, of the first tool that beekeepers can have, which is the genetics. So this is my favorite subject, so you can stop me anytime, and hopefully I don't get carried away with this. So genetics is the first line of defense that the beekeepers have. Before cultural methods, before treatments, the beekeeper can choose the stock and their queens. There are a number of advantageous traits that the beekeepers can include in their selection program. They can be a good tool to reduce the number of treatments or have more options for the treatments to complement an integrated pest management. We have successful programs in North America, for example, the tracheal mites. And there's various the traits that can be included. The tech transfer program in Ontario they had done a great job assisting beekeepers to select for different traits, for example, honey production, defensive behavior, hygienic behavior. I know now they are including VSH as well. So I'm going to talk a little bit about a research or a project that was done in association with Purdue University, with Dr. Greg Hunt in Indiana, and with the University of Guelph. And it's about using grooming behavior to include that in a stock of bees that they named Indiana mightbiter stock. I don't know if you've heard of them. I like the name. So just a little bit of background. There are some stocks of honeybees that have been bred to be tolerant or resistant to Varroa. And they use, there are limited traits, but there are some that are associated to Varroa population growth. One of them is the Varroa-sensitive hygiene that was developed by USDA, and I believe there's a commercial stock available to beekeepers. And then we have grooming behavior. There are also some stocks that have been developed. One of them is the Indiana mightbiter stock. You have the Russian bees. We have some projects done at the University of Guelph. The challenge with using grooming behavior is how do we measure grooming behavior? If you want to select for a trait, for example, honey yield, what you do is you measure the amount of honey that your colony is producing. If you want to select for a defensive behavior, then you count the number of stings in a flag that you put in front of the hive, and then you have a quantity, a way of measuring the trait that you want to incorporate in your stock. But how can we measure grooming behavior? We know grooming is associated with Varroa population growth, but how can we know which colonies or which queens are better than others? So there are some assays that have been developed, for example, individual grooming, and that's a lot of fun. You put a worker bee in a petri dish, then you put a Varroa mite on top of her, and then you see when she starts grooming, if she's able to remove the mite from her body, if she uses 1 or 2 legs and she's lazy, or if she's an intense grooming. So this is a good way to measure grooming behavior, but it's time-consuming. Then we have mite population growth. You take a sample, or you put a sticky paper in the spring, then 16 weeks later, you do the same, and you subtract the amount of mites from the first count to the fall count, and then you have an idea of the mite population growth. Then there's other ways to do it that are I guess, also a little bit time-consuming, but they have worked. For example, using the sticky papers, you can count the number of Varroa mites that were chewed or mutilated and how severely they were mutilated. And then we have molecular markers, such as neurexin. Based on individual grooming assays, they were able to link, Greg Hunt in Indiana and Dr. Guzman in Guelph, they did some research and they saw that the bees that were grooming more, they had higher levels of expression of this gene neurexin. So we know there's a potential to use molecular markers, although not the only way to do it. So it's a way to complement this approach. So just a little bit of background of the Indiana mightbiter stock. It started in, I think, 1997, and they started with a stock of bees that had DSH, Russian bees, but mostly colonies from beekeepers in the Midwest USA. So they started selecting based on lower mite population growth and the proportion of chewed mites and the severity of the mite mutilations. They compared this mightbiter stock in 2016 with a Carniolan stock, and they saw that the Indiana mightbiter bees had lower mite levels and they had a higher overwintering success. So they kept going with the program. On the other hand, we have University of Guelph doing some research and selecting colonies from Ontario based on grooming behavior, individual grooming behavior, and mite population growth. Just give me one sec. So the objective of this research that I'm talking to you about it was to assess the efficacy of breeding bees based on mite mutilation and to evaluate the potential use of neurexin as a molecular marker to assist breeding selection based on this trait. So in 2017, what they did was they divided some colonies, 9 colonies, so at the end they have 18 colonies, and they introduced 9 Italian mightbiter queens and 9 Italian queens. They put sticky papers and did the first mite count. Then 16 weeks later, they did the same. They put sticky papers and counted the mites. We collected the data, we collected bees, and they sent it to the University of Guelph for molecular analysis. And then in February of the next year, they assessed survival or overwintering success. So just as a brief summary, this mightbiter stock was assessed for mite population growth, the proportion of mutilated mites, the severity of that mutilation, overwinter survival, and the expression of the gene neorexin. And we saw that the mightbiter stock had lower mite population growth compared to the Italian bees. So that was a good result. That was one of the goals of this breeding program. So that was good news. We also saw that the proportion of mutilated mites was higher in the Indiana mightbiter stock compared to the Italian. So that's also good news, because when you see fallen mites in sticky papers, you really don't know if they fell because the bees shook them out of their bodies or because they just died. So having the mutilation, it adds more information about how the mite died, and also the severity of the bite. Indiana mightbiter stock showed a higher severity of that mutilation compared to the Italian stock. So that was positive news, and that matched the objectives of the breeding program. We also saw that neorexin had a higher expression in Indiana mightbiter bees compared to the Italian bees. However, this neorexin did not correlate to other variables, only to the proportion of chewed mites. Although it was not significant, you can see a positive correlation. So that was also good news, and it kind of makes sense because the bees were selected randomly. They were not performing grooming behavior when they were sampled for molecular analysis. So what we can see is that selecting honeybee stocks for the proportion of chewed mites, it showed that it's been working for the Indiana mightbiter stock. We can see a potential of neorexin as a molecular marker, or although we cannot only use that, it has to be a complement, and we're looking more into details about that, maybe not the expression, but the sequence itself, but then I'm getting carried away and talking about that. So we see that there's promising results to breed tolerant bees to Varroa destructor based on these traits that could potentially be incorporated in commercial operations and as a complement or as for the integrated pest management approach. The aim is to reduce mite levels again, and at the end to increase overwintering success. So this brings us to one of the last components of integrated pest management, that is treatments. So we saw the first line of defense that the beekeepers have, which is genetics, all the tools that you can use that do not involve chemicals to control the levels of Varroa destructor based on monitoring. So if you incorporate all these, at the end you have to choose, if you need to treat, which treatment to use. But I'm not going to go into detail about this because it mostly depends on your area, your province, and the regulations where your beekeeping operation is. So just as a general thing, in Ontario we always tell beekeepers, follow the regulations and the recommendations of your province. If you have questions, go to the tech transfer team or the apiary program. Follow the manufacturer's instructions. Always monitor your levels and know your enemy. Know what's going on in your yards. And if you choose to use synthetic acaricides, always consider the potential of or the risk of developing resistance. So again, follow the instructions and the recommendations of your province. And just to finalize, at the Honeybee Research Center, we're working on a breeding program that is based on mite population growth and grooming behavior. We are in the third generation of this selection that began with bees from the Ontario stock, beekeepers in the province. And the aim of this study is to be able to incorporate another trait of Varroa control to commercial operations in the province, and also to understand the mechanisms of Varroa resistance. There are other colleagues at the lab that are working in other approaches, for example, on the development of other treatments to treat Varroa destructor, mostly with essential oils, for example. We had another PhD student that worked with biocontrols. Unfortunately, in theory, everything worked fine, but in the field, she didn't have a lot of success. But there's another approach to think about. And, well, this is our information. The Honeybee Research Center also has videos on basic beekeeping practices and recommendations. And what we always tell the beekeepers in Ontario is if you're going to see something online, just be aware that it might not work in your area, or the recommendations might not apply for your province. So be aware of that and the best management practices that your province or your area is recommending. And I don't know if you have any questions. I'll be happy to answer. Thank you.